In old age, bones become thinner. We showed an epigenetic change in old stem cells that are important for bone maintenance. Restoring the epigenome to a young-like state was sufficient to rejuvenate these stem cells in a petri dish. So epigenetic changes are one of the hallmarks of the aging process. And epigenetics describe the change in activity of our genome, so for instance the expression of genes, without changing the underlying DNA sequence. And one way to do this is, for instance, by modifying proteins, which are called histones, that wrap the DNA. This can control, for instance, access to the DNA to allow transcription to take place. My lab is interested in understanding how the epigenome shapes cell fate decisions. And what we knew is that bones, for instance, they become thinner with age. One of the causing cell types for this phenomenon are called mesenchymal stem cells. So these are stem cells that reside in the bone marrow and can differentiate into bone, but also into fat. And with age, these cell types rather differentiate into fat than bone. And we know that there should be an underlying epigenetic reason for this shift. We started our studies by investigating the accessibility of DNA. And we found that in old stem cells, chromatin is more compact. And what we know is that one particular histone modification, which is acetylation, can change the dynamics between more or less compacted chromatin. And in support of this idea of a more compacted chromatin, we found that in aged stem cells, we have less acetylation of histones. As a final step, we wanted to see if we could rejuvenate our old stem cells. And we used a very simple trick that we know from the literature. So we treated our isolated stem cells in tissue culture with sodium acetate. And what we know is that the cells will take up the sodium acetate and convert into a building block to acetylate histones. And just treating our old cells for 3 days with sodium acetate was enough and sufficient to reinstate a young-like epigenetic state. And more importantly, this also led to a higher differentiation potential of these cells, and we could differentiate these into bone cells as efficiently as young cells. The majority of our experiments were done in mesenchymal stem cells that we isolated from mice. As a very last step, we wanted to see if the effects that we observed were also true in human cells. And to do this, we received stem cells from a collaborator that were isolated upon hip surgery from young and old women. And excitingly, we could observe the exact same effects in human cells than we saw before in mice.